Hi, this video is about DNA basics. Okay, so first of all, just starting off with some basic vocabulary terms when it comes to DNA. A DNA is a type of nucleic acid. That is what the Na stands for in DNA. And a nucleic acid is simply a biological macromolecule. And there are two types. There's DNA and there's RNA. That is composed of carbon, hydrogen, nitrogen, oxygen, and phosphorus, and they carry genetic information. What do I mean by the term macromolecule? Macromolecule just means that it's a big molecule. DNA is a very big molecule. It's extremely long. It's one of the biggest molecules we really know about. And DNA is made up of what are called nucleotides. And here's an example of a nucleotide down here. And a nucleotide is the monomer of a nucleic acid polymer. So what do we mean by those terms? Well, a monomer means that it's a single building block that makes up a bigger molecule. So the DNA double helix, like you see here, right? That is a big, long macromolecule, but it's made up of these smaller repeating subunits that repeat over and over again called nucleotides. And every nucleotide has three parts. Those parts are the nitrogenous base, which you're going to be having these. This is a nitrogenous base you're going to be using to build a DNA model in a bit. They also have a 5-carbon sugar. Here's an example of one of those. You'll be using those when you build your DNA model. And then here is a phosphate group. And the phosphate group is the last part. And those three pieces fit together to make a nucleotide. So as a reminder, the nucleic acids, like DNA and RNA, they are polymers, poly meaning many, many pieces put together to make a big molecule. And then nucleotides, those are the monomers. Mono meaning one piece. All right, a little bit of comparison and contrast between DNA and RNA. It's not that important now, but I want to prime you for it. We will be learning more about RNA later this school year. So DNA you're familiar with, right? Deoxyribonucleic acid, that's where the name comes in. And DNA is a double-stranded helical molecule, which carries genetic information, and it's often called like the blueprint for life. RNA, another type of nucleic acid, is a single-stranded nucleic acid. It's a little bit different. Its main purpose is being used to pass along genetic messages, but there are some different types of RNA that each have their own particular functions, and we'll come back to that later on in the school year. Just want to make sure you're aware of some of the basic differences between DNA and RNA. RNA standing for ribonucleic acid. All right, some basics of the structure of DNA. So these green dots here, this is what's called a ladder model of DNA, and the green dots are phosphate groups, right? Those are these guys right here. They're your yellow and red pieces they're going to be using in your model. These orange pentagons are the 5-carbon sugars, which in DNA, that sugar is called deoxyribose. As a reminder, deoxyribose sugars. These in the middle are your nitrogenous bases, right? In this example, this C one here, that's cytosine. That's one of your bases here. And then you have a nucleotide, right? As a reminder, that's all three of these pieces put together. That's one of your monomers. And you see how that repeats, right? You repeat base, sugar, phosphate, base, sugar, phosphate, base, sugar, phosphate, base, sugar, phosphate, up and down the different sides. This part down here along the side, we call that the phosphate sugar backbone. So when you see like a DNA model, you see those strands twisting around each other. Those strands are twisting around. Those are the phosphates in the sugar, just repeating every other, right? And then the bases are on the inside, and they're paired up. Speaking of pairing up those nitrogenous bases, those nitrogenous bases, there are 4 of them in DNA, but there's actually a 5th one that we talk about in nucleic acids that we'll come back to later this school year, like I said. The nitrogenous base is the variable part of DNA. It's the part that can change. It's kind of like the letters of like the genetic alphabet, right? If you can kind of think of DNA kind of like a code, it has its own alphabet, just like a language has its own alphabet. But this alphabet only has 5 letters. And in DNA, there are actually only 4 of them. And those are cytosine, guanine, adenine, and thymine, right? These 4. But then there's also another one, and that's uracil, which is only used in RNA. And we use the letters G, C, A, T, and U to represent these different nitrogenous bases, the part that can be different between the different nucleotides. One thing you want to know is that these top ones, the larger ones, are called purines. These are called the purine bases, and the smaller ones are called pyrimidines. A couple of helpful hints to help you kind of remember these. Pyrimidines have Y in the name, thymine and cytosine, except for uracil. Uracil doesn't, but uracil takes the place of thymine. So if you remember that thymine is a pyrimidine, then you should know that uracil is a pyrimidine. Whereas the guanines and adenine, right, the purines, those don't have a Y. So pyrimidine has a Y, and thymine and cytosine have Ys. Purines are the larger ones. They have 2 rings, okay? And pyrimidines only have 1 ring. The way that I remember that is when I think of a pyramid has 1 point at the top, right? There's 1 point at the top of a pyramid, just like there's 1 ring in the pyrimidine bases. And the way these pair up is purines always pair up with pyrimidines. So let's talk a little bit about those base pairs. A base pair is just when you have those 2 bases going together. So those nitrogenous bases are held together by what are called hydrogen bonds, which are a weak type of bond. And the rules are that guanine always pairs with cytosine, and the adenine always pairs with thymine in DNA, but not in RNA. In RNA, we use uracil instead. Once again, we're going to come back to RNA later this school year, so keep it in mind for later. And this whole principle, the base pairing rules, were discovered by a scientist named Erwin Chargaff, and they're known as Chargaff's rules. And they say that for every C, for whatever amount of cytosine you have, you're going to have that same amount of guanine in any DNA sample, right? And the same is true for adenine and thymine. And this principle was really important in the discovery of the structure of DNA. Basically, as people are trying to figure out what DNA looked like and what it was and what it did, this scientist Chargaff, he discovered that when he was doing his chemical analysis of DNA, didn't matter what DNA sample he was looking at, whether it was a frog or a person or a bat or a tree or whatever it was, the amount of cytosine always matched the amount of guanine, and the amount of thymine always matched the amount of adenine, and that's because they pair up like this. Between C and G, there are 3 of these weak hydrogen bond attractions, and between A and T, there are 2. The way that I remember this is that cars park in garages, C and G, and apples grow in trees, A with T. Cars and garages, apples with trees. All right, so quick refresher, a little review over the DNA and RNA, some of their basics and some of the differences between them. DNA stands for deoxyribonucleic acid. It is double-stranded. It has deoxyribose as its sugar. Its 5-carbon sugar is deoxyribose. And it uses the bases C, G, A, and T. RNA, a little bit different. It is only single-stranded, and it uses ribose as its sugar, hence RNA instead of DNA. And it uses the bases C, G, A, and U. All right. After watching this video, there is a mastery check over the DNA basics, and then you'll be moving on to the part of the procedure where you're going to be dealing with these models. So you'll need to come up and you're going to need to grab one of these boxes, the DNA Discovery Kit boxes, and you're going to start by drawing models of the different components of DNA in your lab journal, and I'll be checking those. And then after that, you'll be moving on to building your models. Now, importantly, when you build your models, you have to build nucleotides first. Anytime you build a polymer, you have to have the monomers first. So don't do anything with the magnets until you've done the pegs and the holes, right? So for example, here's a little half circle, half moon peg there. That peg fits into this hole on the sugar, right? Fits right in there. So you can put those together. And then the other peg, right? I've got this kind of trapezoid peg here on the phosphate. That peg will go into the trapezoidal, trapezoidal hole on the sugar, and there's one of my nucleotides, right? It's got its phosphate, it's got its sugar, and it's got that nitrogenous base. This one, once again, cytosine, right? You have to build these first. So pegs and holes before you do magnets. Once you have all of your nucleotides built, then you use the magnets to connect those together to make your DNA model. Of course, if you have any questions or need any help, I'll be here to help you out. Good luck, everybody.